Guys, my name is Mr. Mpepu and today's date 19 September 2024 and the time there 39 minutes past 11. With that being said, guys, Sanbonani, Dumelani, Avsheni, hello, Nda. Guys, I personally welcome you to our Life Sciences Revision Classes Term 1 Week 4. Under meiosis, right? So today we are at week 4. We are done with our DNA, the code of life. We are moving to meiosis, the structure of chromosomes, the associated terminology, process of meiosis, importance of meiosis. Hopefully we can cover this in less than 10 minutes or 10 minutes. So let's move on, guys. Thank you for being here. If you are new here, don't forget to subscribe, guys. Don't forget to like, don't forget to comment. If you are a returning student, guys, don't forget to like and don't forget to comment. Thank you for pushing this movement with me. Thank you for joining me. Thank you for choosing me. Thank you for tuning in, guys. Thank you. Let's move on. Let me not waste your time. 10 minutes, start. Meiosis, guys. Meiosis in grade 10 we did mitosis, guys. We did mitosis where a cell was divided to form two identical daughter cells. And then now in grade 12 we are learning meiosis, where this is the special type of cell division that halves the number of chromosomes in the 4 genetically different haploid daughter cells formed from a one diploid cells, right? The meiosis is very important because the haploid cells are produced and then the genetic variation occurs, and also it overcome the doubling of fertilization on chromosomes, right? So we have another terminology that we'll have to cover fast, but we have the chromosome. A chromosome is made out of a DNA and a protein, and then it's found in most living cells, and then this here is the chromosomes, and then it also have the chromatid made of one chromatid. When they are replicated, they are chromatids. These are the chromatid here, right? And then they are joined at the region called centromere, and then when they are together like this, when they have the same shape, same size, and having the similar gene, they are called homologous chromosomes, right? And then when they come together during prophase, they make a physical contact, and then they are called bivalent. You know about this, you remember about this. Let's just jog our mind and do a recap. And then we also learn about unreplicated chromosomes and replicated chromosomes. As you can see, we are conducted by our centromere there. When we have one centromere with 2 chromatids or 4 chromatids in total, we can now conclude that our chromosomes have already replicated. When we only have one centromere and 2 chromatid, it is our unreplicated chromosomes. And then all this replication takes place in our interphase, and then we also have the diploid and the haploid, where the 2 complete cell of chromosomes, we call them the diploid. And then when that, as you can see here, we have the centromere and the centromere to show that these 2 are the 2 sets of chromosomes, and we call them the diploid. And then when we reduce one, we have our haploid, which is the 1 complete cell of chromosomes in a single cell. And then we also have the gene there, a gene which is this. We also have a gene which is the a code, a segment of DNA in a chromosomes that code for a particular characteristics. We have a chromosomes, and then we also have a small part there called a gene that code for a specific characteristics. We also have the centrosomes, only found in an animal cell, that split and form a centriole, and then this centriole moves to a different pole and produce the spindle fibers. And then we also have the crossing over that takes place during the prophase 1, and then where a genetic material are being exchanged. And then in this process we see that the place where these 2 chromosomes, these homologous chromosomes overlap, we call it chiasma, or when there are many, we call them chiasmata. And then as you can see there, our genetic chromosomes, or the chromosomes, they have exchanged our genetic information. And then we also have the karyotype, which is the set of chromosomes from pair number 1 up until pair number 23. And we also have our autosomes, which is the pair from pair number 1 up until pair number 22, which all control the appearance and the structure and the function of the body. And we also have the gonosomes, which are the sex chromosomes, pair number 23, guys, as it can be X and X and X and Y for the male. And then we also have 2 types of cell, the somatic cells, which are the body cells, and then we also have the sex cells, which we call them the gametes. And then we also have the process called non-disjunction, where a chromosome fails to separate during a separation during the meiosis, right? So when the chromosomes, they fail to separate, we call the process is called non-disjunction. And then we have 2 types or 2 terminologies, karyokinesis and cytokinesis. Karyokinesis, this is the division of a nucleus, and cytokinesis is a division of our cytoplasm to form 2 daughter cells. And then we move from there, we go to the significance of DNA replication for meiosis. Why the DNA replication is very much important for meiosis, so that to ensure that the sharing of hereditary hereditary material by all daughter cells that will be formed. So that while you start with one chromosome, and then that chromosome is going to, that DNA is going to replicate, and then after replicating we are going to divide this in two so that each daughter cells can have the hereditary material with it. So we are moving on to the process of meiosis, and then as we can see, this process of meiosis is very much important, so that the chromosome number is not doubled when fertilization occur. Instead, when two haploid chromosomes fuse, the diploid number is then restored, right? Then the meiosis ensures that the number of chromosomes within the species remains constant from generation to generation. In animal, meiosis occurs in sex organs called ovaries and testes, you know about this, to produce gametes in the process called gametogenesis. And then in plants, we mostly see them in mosses and ferns to produce spores, and then in angiosperm or the flowering plants we see it happening in the anther and the ovule. And then we move on, but the note there is just that they're telling us that in our human body we have 46 chromosomes and then in 23 pairs, so it's going to be practically impossible for us to represent these 46 chromosomes, so we're only going to use 4 chromosomes, or we only use 4 chromosomes in our illustration of our meiosis. And then in this process, meiosis undergoes 2 division, which is meiosis 1 and meiosis 2. Meiosis 1 being the division with being the reduction division, where 46 chromosomes is divided into 23 chromosomes, right? And then from there, this 23 also divide the chromatid here, and then we'll see when we move on. You know what I'm talking about. The first meiotic division, although meiosis is continuous process, the event are placed into phases for convenience, and then we are going to use our mnemonic PMAT, prophase, metaphase, anaphase, and telophase, right? P1 and 2, right? And 2, prophase 1, prophase 2, metaphase 1, metaphase 2. But in prophase 1, same thing happens with mitosis. A nucleus membrane and nucleus is going to disappear, as you can see, starting to disappear, and the chromosome or the chromatic network, they are going to condense and form a visible chromosomes. And then from there, the centrosomes is going to split into 2 centrioles and move apart, forming a spindle fibers, and then the inner chromatin network of homologous chromosome. The process that occurs in our prophase 1, we say that we have the contact there, which called chiasma, where a process of crossing over is taking place, and then the chromatid segment breaks and exchange, and then resulting in the exchange of genetic material. And then the process is called crossing over, and it brings about a variation. As you can see, there's the process of crossing over, and we also have the metaphase 1, where these 2 homologous chromosomes, they are arranged at the equator, and from there we have to understand that they are randomly arranged there, also to bring our further a variation there. And then from there we have anaphase 1. With anaphase 1, the whole chromosomes in this anaphase 1 is going to be pulled to the opposite poles, contracting of a spindle fibers, right? And then this separate the homologous chromosomes, each one pole and each to the another pole. And then from there we have the telophase 1, where the cytokinesis is taking place and then splitting the mother cells into the 2 daughter cells. And then we started with 46 chromosomes, and then we are ending with 2 cells, both of them having 23 chromosomes. And then we move on to our second mitotic division. We move on to our second mitotic division from prophase 2. Prophase 2, same thing is happening. The nuclear membrane, the nucleolus is going to disappear, the centrosome is going to split into the centrioles, and the spindle fiber is going to be formed, and then the chromosomes are not in pairs this time. Actually, we have 23 now, and then this one is not a pair, it's a single chromosome, 1111111, right? And then from there, this 1111 chromosome, or the single chromosomes, they are going to move to the equator under the metaphase 2, right? And then when they are at the equator, and then which the chromatids face, which the pole is totally up by chance. And then also here we're also talking about the random arrangement, right? Each chromosome become attached to the spindle fiber, ready for anaphase 2. And then in anaphase 2, our chromosomes or our chromatids, they are going to be pulled to the opposite direction. As you can see now, this we are only concentrating on our chromatid, right? And then now they can be pulled to our opposite direction using our spindle fibers. And then from there we move to our telophase 2, and then in our telophase 2, we can see that our cytokinesis is happening. Our cytokinesis is splitting the cells into the 2 cells, and then we started with 1 cell, and then it is resulted into 2 cells, and then these 2 cells here, they are going to divide, and then we end up with 4 genetically different cells. 1, 2, 3, 4 genetically different daughter cells, right? And then the nuclear membrane will form around the unreplicated chromosomes at each pole. As you can see, our nucleus membrane, they are being formed, and then the nucleoles there, they are being formed. And then as we can move on, we see the importance of as a meiosis 2 took place in 2 cells, there will now be 4 daughter cells. These cells will be haploid genetically different from each other, and as you can see there, they've already given us our menu. What would you call them? What? Mnemonics. PMAT, right? Prophase, metaphase, anaphase, telophase. And then why is crossing over important? To bring upon a genetic variation, right? And then by the formation of new genetic combination, and then being genetically different from your parents and your siblings. For you to be different from your siblings is due to crossing over, the exchange of those genetic material. And then the importance of meiosis, the production of gametes, the halving of chromosomes from diploid to haploid, so that the chromosome number remains constant from generation to generation. And the mechanism to introduce genetic variation, our crossing over and our random arrangement during metaphase 1 and metaphase 2. And then that's it. And then this is the picture showing our random arrangement. My time is up, my alarm is off. As you can see, guys, metaphase 1, metaphase 2, our random arrangement, these chromosomes here, we can start them like this, and then you can see that the other one can decide to be this direction, the other one can decide to face this pole. But also we can also have or there, this one can can decide to face this pole, and then the pink one can decided to face this pole. Also, you can also anyhow, it's just a random arrangement. They are being arranged randomly at the equator. So thank you guys, thank you for tuning in, thank you for choosing me. My name is Mr. Paper. Until we meet again tomorrow, when we are doing differences between meiosis 1 and meiosis 2. That was our revision for today. Thank you for choosing me. Until we meet again, guys, thank you. My birthday 4th of October, but don't forget. Thank you. Peace. Let's